Okay, so good evening, guys. Today will be the last lecture for the course of Introduction to Cell Biology. So I request everybody to whatever doubt they have, you must ask in this lecture. Okay, so to start with, first we'll go through the course recap. What exactly has been done in the course of that 8 weeks. Okay, so starting with in week one, we actually started with the introduction, that is the central dogma of the molecular biology, how the genes work, and how the information is actually transferred from DNA to protein. Hmm, Sorry, I don't have the pad today, so I'll try to write all the answers. Okay. Then from that we moved on to the structure of DNA and how it was deduced, what are the different components of that structure, the nitrogenous bases and the phosphate group, followed by nucleotide, nucleoside, and the Chargaff's rule, the complementarity of the DNA, and followed by the replication, how a DNA molecule is replicated in a molecule, in a cell. Okay. Then moving on to week two, we discussed how a gene is basically expressed in a cell through transcription and translation. Okay, and how after forming the from DNA to RNA, that is transcription, and from RNA to proteins, that is translation, and followed by the folding of that particular protein to form a functional counterpart, so that the phenotypic rescue or phenotypic function can be seen. Okay, then in week 3 we continued with the protein folding part and basically described different other biomolecules like carbohydrates, lipids, and enzymes. And then we started with the genetics, basically the Mendelian genetics. We discussed about different laws, the law of law of dominance, law of independent assortment, and law of segregation that the Mendel described. And then we followed with the non-Mendelian genetics, that is linkage and recombination. So that is the modern genetics that we study. Then in week 4, we started with the introduction to microscopy and different components of the microscope that we use daily and in the labs, and the modern microscopy techniques that are being followed here. Okay. Then continued with the introduction to the cells, the prokaryotes and the eukaryotes, and then we moved on to the endosymbiont theory that basically defines the origin of the two major organelle in the eukaryotic cell, that is mitochondria and the chloroplast. Okay, then moving on to the second part of the course, that is from week 5, we started with different components of the cell. So that is cell wall, the outer covering of the bacterial and the plant cells, the cell membrane, and the cytoskeleton. Okay, then the cytoskeleton is basically the skeleton of the cell. So how a cell maintains its shape and integrity inside the cell. So that is being introduced in the cytoskeleton. Okay. Then moving on to the week 6, that continued different components of the cytoskeleton. So we discussed about the actin filaments, that how a cell maintains its shape using the actin filaments, and the intermediate filaments, that is the size of these filaments is between microtubules and the actin filaments. So and one of the major key intermediate filaments is present in the nucleus, that is lamins. Okay, then moving on to the major component of the cell, that is the endomembrane system. So this comprises of around 40% of the total membrane components. Total membranous component of the cell, and it includes mitochondria, Golgi body, then lysosomes, vacuole, and then all the secretory proteins that are secreted out of the cell. So the major one is endoplasmic reticulum. I'm sorry if I said mitochondria. It is not mitochondria, it's endoplasmic reticulum. Okay. So that is the major component of the endomembrane system. And how the proteins are translocated to the membranes and to outside of the cell, that is a secretory part of the secretory proteins of the cell. Okay, so this we discussed in week 7, and moving on to week 8. In week 8, we discussed majorly about the nucleus part, how different components of the nucleus are maintained, the euchromatin and heterochromatin. Followed by we move to the last part of the series, that is the cell division, how a cell divides, what are the different phases, and how a new cell is formed from the pre-existing cells. Okay, so these were the components of the whole lecture, and if you guys have any doubt, we can discuss right now from any lecture. Or else we'll start with some of the questions that I have prepared from whole of the course. And meanwhile, if you guys have any doubt, you can ask in between. Okay, so moving on to the first one, that is in sea urchin DNA, which is a double stranded DNA, 70% of the bases were shown to be cytosine. The percentage of other bases present in the DNA is. So, what can be the correct answer? You can apply Chargaff's rule here. Okay. Any more answers? Okay. So here we have given that the cytosine is around 17%. Okay. So cytosine will be equivalent to G, that is guanosine. Okay. So guanine, sorry. So because cytosine pairs with guanine with the three hydrogen bonds. Okay, so the number of guanine residues would be also 17%. Okay, now the uh, as per the Chargaff rule, A plus T, sorry. So what will happen out of 100%? Just 1 minute. sorry. So, like out of 100%, that is the number of bases present in the double stranded DNA, C and G combining will give us 34%. Okay, so out of 100%, 34% is C and G. So the remaining will be 66%, the left ones. Okay, so out of the 66%, the A would be half, that is 33%. And because it would be binding to T with a double bond, double hydrogen bond, so this would be equal to 33%. Okay, so the C and G would be equal 17% and A and T would be equal 33%. So the correct option would be option number 1. Okay, so do anybody has any doubt? Okay, so let's move on to the next question. How many number of replicon is found in E. coli? So what can be the correct answer? Okay, any more answers? Any more answers? Okay, yes. So the number of chromosomes present in an E. coli is how much? This is an open question to all. Yes. So there is only one chromosome that is present in a prokaryotes. Okay, so that is actually considered as a one chromosome. Okay, so because there is only one DNA molecule that is circular in nature in case of prokaryotes. Okay, that is considered as the main chromosomal DNA or the nuclear DNA that we call it. Okay, or the genomic DNA. Then from this one chromosome that they have, they only can replicate through a single origin of replication. Okay, so this single origin of replication is now here termed as a single replicon. So they consist of only one replicon, and in case of the eukaryotes, especially in case of humans, there will be multiple replicons. Okay, so the correct answer is option 3. That is, it has a only single replicon. Okay, so any doubts in this question? Okay, so let's move on to the next question. DNA replication is bidirectional and anti-parallel. Just one minute. So DNA replication is bidirectional and anti-parallel. Which of the following statements regarding the DNA replication is false? So what can be the correct answer? Any more answers? Okay. So the correct answer is option 4, that is none of the above. So all the options that are stated here are correct. Okay, how? So this is an example of the initiation of replication in prokaryotes that we see generally see. So what happens is the DNA synthesis, that is addition of nucleotide, occurs from the 5 prime to 3 prime direction. So yes, this is correct. DNA synthesis is semi-continuous with continuous leading strand and discontinuous lagging strand. Yes, this is also correct because this in one of these strands it is actually leading. So in this particular case, it will be leading. That means a continuous stretch of the replication for this, for example. Okay, so this is the leading strand that we can see here. This one. Okay, so this is the leading strand because here no breaks are being formed. Okay, but in the other strand, different particular fragments that are called as Okazaki fragments are being formed because of this opposite direction of the DNA replication. Okay, so this is called as discontinuous replication that we can see here. So both occur that is why it is called as semi-continuous. Okay, and the last one, the synthesis of leading and lagging strands occurs simultaneously. Yes, this is also correct because the DNA polymerase has two domains, two polymerase domains. Okay, so one of the domain actually synthesizes the leading strand, and the other one synthesizes the lagging strand. So both the replication occurs simultaneously. Okay, so this option is also correct. So the correct one is none of the above. That means all of the options are correct. Okay, so any doubts in this particular question? Okay, so let's move on to the next question. The following DNA strand is used as a template for transcription. So which of the following RNA strands will be produced? So what can be the correct answer? Mhm. Please read the question carefully. We have this as a template strand. Okay, so what will be the sequence of the RNA? Any more answers? So RNA synthesis also occurs from 5 prime to 3 prime direction, right? So if this strand is used as a template strand, then from 5 prime to 3 prime direction, the RNA sequence would be G, C, A, then U, U, followed by C, G, C, C, G, and then A. Okay, so this would be the sequence of the RNA, and sorry for my handwriting. Okay, so this is the sequence that we'll get from the RNA. So that is option number 4. Okay, and if it would be a sense strand, that means the sequence of that particular strand would be same as that of the RNA, but the T will be replaced by U. Then the RNA sequence would be this this one. Okay, the option number A. So any doubts in this particular question? Okay, so let's move on to the next question. A normal E. coli cell produces a polycistronic mRNA for genes X, Y, and Z. This mRNA is significantly shorter in a mutated E. coli cell. Which component of the transcription mostly likely contains an altered sequence in the mutated cell? So, what can be the correct answer? Okay, any more answers? Okay, so a normal E. coli cell actually produces a mRNA that contains three genes. Okay, this is X, Y, and Z. So this is a normal scenario where a polycistronic mRNA contains all these genes, and but in a case of the mutated cell, what happens? There is a shorter RNA. Okay, so shorter RNA is formed. That means either Y or Z is not there. Okay, so consider that Y is not being produced here. So only X is contained in the mutated mRNA. Okay, so this is mutated. Now, what can be the possible reason that this mutation is happening here? So this is what the question is. So what can be the correct answer? Okay, any more answers? Okay, yes, the correct answer is option number 2, that is a termination region. Okay, So first of all, in a E. coli cell, because it's a prokaryote, it doesn't have a intron region. Okay, so it doesn't have a intron region. So the first option is neglected. First of all, okay. Then, if the mRNA is being shortened, then the promoter region will not have any role in this. Okay, if the promoter would have been mutated or there would be any problem associated with the promoter, either the mRNA will not be produced or it will be produced in more number. Okay, so this reason is also neglected. Then a spacer region. So a spacer region will not be a correct answer because in that this case there is no spacer region. Okay, in case of polycistronic mRNA, there is no spacer region present. The only region that can have the mutation in this case, because it is forming a shorter mRNA, that is a termination. Okay, so that means a termination region is actually being created, or the site of termination is actually being created in between X and Y gene. That's why the RNA is being terminated here. Okay, so that can be the possible reason from the given options. So, any doubts in this particular question? Okay, so let's move on to the next question. A mutated eukaryotic cell contains a mutation in the middle of an exon in gene Q. What will be the most likely result of this mutation? So, what can be the correct answer? Okay, or anybody else? Any more answers? Okay, so because the eukaryotic gene have certain intervening sequences that we call it as introns. Okay, so these would be the exons of this particular gene Q that is in question. Okay, and one of the exons have a mutation. So this is the mutation that is being say, uh, told in the question. So out of these options, if we see, if there is a mutation in the exon, then the amount of transcription of gene Q would be greater than normal. So if there is any mutation in the exon, then there would be no effect on the transcription levels. Okay, if there would be any mutation in the promoter or the enhancer region, then we would have speculated that the transcription of the gene might be normal, greater than the normal. Okay, so this option is wrong. Then mRNA produced from gene Q would be longer than the normal. So if that would be the case, because here we the splicing inability is being talking is being taken into consideration. So if there would be any mutation in the introns, that would actually hamper the splice site, that would result in a longer mRNA than the normal scenario. Okay, so this option is also false. Then the enzyme produced from gene Q would have less activity than normal. So because an exon that have this mutation, might this mutation will be present at the active site of the enzyme, because this exon will lead to the protein finally. Okay, so this will lead to the protein, and if there would be any mutation in the active site of the enzyme, then that would hamper that enzyme activity. Okay, so this option can be correct. Then the breakdown of the mRNA from gene Q would be slower than normal. So this option would also be false, because for breakdown of this mRNA, there are some signals that are associated with it, and those signals are not present in the exons. Okay, so the correct answer would be option number 3. Okay, so any doubts in this particular question? Okay, so let's move on to the next question. So this is the next question. Using the codon chart, determine the sequence of amino acids that is produced by the translation of the given RNA strand. So what can be the correct answer? हाँ, बोलो. Hmm. Okay. Any more answers? So let's start with understanding this codon chart and how to read it. Okay. So as we can see, so codons are a basically three letter sequence. Okay. So codons are basically three nucleotide letter sequence that actually codes for a single amino acid. Okay, and because there are three letters associated with, there will be different amino acids that will be associated with it. Okay, so the last uh, nucleotide defines the different amino acids. Okay, so what will happen? The first letter would be written on the left side, the second letter would be written on the middle, and the last one would be written on the right side. Okay, and as we combine these three, we'll form one codon. Okay, that codes for one particular amino acid. So, for example, here we have AUG. Okay, so AUG codes for methionine. Okay, and there is two specialities of this particular thing. AUG only codes for methionine. There is no other codon that codes for methionine in a general scenario, and this is considered as the start codon. So, in most of the cases, in every case, AUG will be the start codon, and it will code for methionine. Okay, then comes the other codon, that is CCA. Okay, so C will come here, then in this case C, and this is A. So this is the second codon, and this codes for proline. Okay, so this is proline. Then we have the other codon, that is CAC. Okay, so in this case C will come in the second quadrant, A will come in the third quadrant in the middle one, and then C would also come in the second quadrant. So this codes for histidine. Okay, so the third codon would be of histidine. Then we have AGG. So A, third quadrant, then for the middle one it would be the last one, and for the last one it would be the third. So this is AGG, and this codes for arginine. Okay, so this codes for arginine. And then we have GGA. So G will be in the fourth quadrant, here G would be here, and this is A. So this codes for glycine. Okay, so this codes for glycine. So the correct sequence would be methionine, proline, histidine, arginine, and glycine. So that is being mentioned in the third option of the question. So the correct answer would be option number 3. Okay. So any doubts in this particular question? Okay. So let's move on to the next question. The table below describes the effect of antibiotics on protein translation in bacteria. So what can be the correct answer? You have to match the following basically. Okay. Any more answers? Okay. So these antibiotics basically inhibits different parts in the translation of the bacteria. Okay. So as we see, bacteria has ribosome that is 70S, and this 70S contains 50S and 30S subunits, and it also has three ribosomal RNAs, that is 23S, 16S, and 5S rRNA. Okay, so different antibiotics bind to different portions of this particular translation mechanism. Okay, so for example, erythromycin is an antibiotic that basically affects the 23S rRNA of the 50S subunit of the ribosome. Okay, so 23S rRNA is the peptidide transferase enzyme that helps in formation of the peptide bond. Okay, so this 23S rRNA is inhibited by the antibiotic that is erythromycin. Then chloramphenicol, it basically inhibits the 50S ribosomal subunit, that is basically the larger subunit. Okay, so this larger subunit has the elongation factors. Okay, so this elongation factors is basically inhibited by chloramphenicol. Then tetracycline, it inhibits the smaller subunit of the ribosome, that is 30S, by interfering in the binding of the amino acid tRNA. Okay, so it basically inhibits the binding of the tRNA that is carrying the amino acid. Okay, so this is tetracycline, and then we have streptomycin that basically binds to the 16S rRNA subunit of the 30S subunit of the ribosome. Okay, so A first, B second, C third, and D fourth. So the correct answer is option number one. Okay, so any doubts in this particular question? Okay, so let's move on to the next question. The mRNA segment given below is complete to be translated into a polypeptide chain. Study the diagram and answer the following questions that follow. So, what can be the correct answer? Okay, so if we see the question, so because it's 5 prime end, this would be the start of the mRNA. Okay, from here it would be the start, and this is the 3 prime end, so this would be the termination. Okay, so if we have to check, A codon will be the start codon of the peptide chain. So for the peptide chain, so this would be none other than A, U, and G. Okay, that codes for methionine. And B would be the termination codon. Okay, so as we have seen in the last slide also, so the termination codons are three, that is UAA, UAG, and UGA. Okay, so these three are the termination codons. So if we see here, either it can be UAA, UGA, or UAG. So the correct answer is option number 3, because the start codon would be AUG and the stop codon would be UGA. Okay, so any doubts in this particular question? Okay, so let's move on to the next question. Does polysaccharides reduce U2 plus or Fe2 plus ions? So what can be the correct answer? Okay, any more answers? Okay. So by reducing CO2 plus or Fe2 plus ions, I mean that whether polysaccharides can be called a reducing sugar or not. Okay. So what happens is, as a polysaccharide chain is produced, okay, so this is the growing chain of the polysaccharide. One end would be the end that would be having a free functional group, okay, and the other end would be having the non like there would be no functional group. Okay, so because in most of the polysaccharides, for example, in case of glycogen or or in that thing cellulose, there is one end that doesn't have any functional group, and the other end that might have a free functional group. Yes, yes. So they will not be considered as a reducing agent, but there is, like, the one end of the polysaccharide would be reducing and the other end would be not. Okay, so because one end have a free functional group that can actually carry out the further reaction. Okay, so if we apply any of the reducing agent, like reducing test to the polysaccharides, they will give the test. Okay, because their one end is actually reducing and the other end is not. So they will actually reduce this particular thing. So, but they will not come under the category of reducing or non-reducing sugars. Okay. So any doubts? Okay. So let's move on to the next question. In peas, a tall yellow seeded plant is crossed with a homozygous short green seeded plant and it yields 203 tall green seeded plants and 199 short green seeded plants, 207 tall yellow seeded plants and 192 short yellow seeded plants. Okay, the most likely genotype of the tall yellow seeded parent is, so what can be the correct answer? Okay, any more answers? So, any more answers? Okay, so there can be two possibilities here. Yes. Okay, so here we have a tall, yes. So here we have a tall and yellow seeded plant that is crossed with small or short green seeded plant. Okay, so from this case, we can say that 203 seeds were tall, and so there would be 203, that is the first case, that have tall and green seeds. Okay, so this will be small y. Then 199 is short and green seeds. Then 207 are tall yellow seeds, and then the last one is short and yellow seeds. Okay, short and yellow seeds. Okay, so I would like you guys to draw two crosses. One is this one. In this would be heterozygous for both the genes, and the other one is this one. Okay, so from the first cross, because it's heterozygous, it will form four gametes. Okay, so for the first one, it will form four gametes, that is capital T capital Y, capital T small y, then the last one would be small t capital Y and small t small y. Okay, and from the recessive parent, we will have only one gamete, that is small t and small y. Okay, so if these particular gamete fertilizes with this particular gametes, then four type of offspring would be formed. Okay, as there would be mentioned here. So the parent that have a genotype or that have a phenotype of tall and yellow seeds, that would be heterozygous for both the conditions. Okay, and if the parent would be homozygous, then only one gamete will be formed, that will be capital T capital Y, and from this one also single gamete, small t small y, and if they both fertilize, then that would lead to capital T small t capital Y small y. So only one type of offspring would be formed from that case. So that's why the genotype of the parent would be capital T small t capital Y small y. Okay, so the first answer would be correct. So any doubts in this particular question? Okay, so let's move on to the next question. If the progeny of a cross small a capital B is crossed with capital B small b is test crossed. The following genotypes were observed. So what will be the recombination frequency between the total uh, between the two genes? So what can be the correct answer? Okay. Any more answers? Okay, any more answers? Okay, so to solve this kind of questions, first we have to check what is the total number of offsprings are. Okay, so the total number comes out to be 1075, if I am not wrong. Uh, yes. So what you have to do is you have to add these numbers that are given here. Okay. So this is the total number of offsprings that is being produced from this particular organism. Okay. So this is comes out to be 1075. Right. Now the formula for the recombination frequency is basically the percentage, the total number of non-parental offsprings divided by total number of offsprings into 100. Okay, so this is the formula for the recombination frequency. So this is the non-parental offspring, the total number of non-parental offsprings divided by the total number of offsprings that have been produced in that litter, and that is being multiplied by 100. Okay, so what will happen now? The non-parental offsprings are the ones that will be less in number. So this is the criteria for determining the non-parental offsprings. Okay, this is 120 and this is 135. Okay, and the logical reason comes from here. So the original genotype of the parent was small a, capital B, and capital A, small b. Okay, so these were the original genotypes of the gametes. So that means these two are linked and these two are also linked, right? And if we test cross, that means the gamete from the second parent would be small a, small b, right? So from this, four types of offspring will be produced. That would be capital A small a, capital B small b. Okay, so in that case, what will happen? This would have been recombined. Okay, because this gamete would be linked. So this is a, because of the crossing over, this would be a non-parental offspring. Similarly, small a and small b will be from two different chromosomes, two different homologous chromosomes. So they would have recombined and this would form a non-parental offspring, that is small a, small a, small b, small b. Okay. Now if we see here, capital A small b and small b would form one. This is one of the offspring because here the capital A and small b are linked. Okay, and this would be also one of the parental offspring because small a and capital B are linked. and this comes from one of the parents. Okay, so that's why these two are the parental offsprings and the other two are the non-parental offsprings. Okay, till now the explanation is clear. Okay, so if we now add this particular non-parental offspring, that means it would be summed up to 255. Okay, and if we divide it by 1075, that would yield us about, okay, just 1 minute. So this would yield 23.72%. Okay, so the correct option is option number 3. Okay, so what happens is, in case of linkage, I'll explain you in this way. Okay, so these are the parents. So these yielded a offspring that have a genotype small a, capital B, and this is capital A and small b. Okay, so this is the genotype of one of the parents that is being produced from the parents that have this offspring, because these genes are linked. Okay, and the other, because we are doing a test cross here, this will be small a, small b, small a, small b. Okay, so when the gametes would be formed from this case, this would be small a, capital B, capital A, small b, because no crossing over happens in between these two. If the crossing over happens, that means this will be exchanged with this and this will be exchanged with this. Okay, so now the non-parental gametes that would be formed would be small a, small b, because the crossing over happens. So this a will be joined to this b, small ones. Okay, just 1 minute. And the other non-parental offspring would be capital A and capital B, because of the crossing over between this one and this one. Okay, so these are the parental gametes that have been formed from this one, and these are the non-parental gametes that have been formed from the crossing over. Okay. Okay, so is this particular explanation correct? Okay, and yes, how it is coming 40? Because if we divide 255 by 1075, that would yield us around 23%. Okay, if we multiply it by 100. Can you please be clear about the question? I'm not getting it. So what uh, I am guessing from your question is how a recessive character is being expressed in an organism, right? Or how any gene can express a recessive character. So there are two kinds of characters that have been studied. One is the wild type or the dominant one, and the other one is the recessive one. That you can call it as a mutated version. Okay. So the wild type or the dominant one is frequently found in nature. Okay. Or the nature has accepted that this particular trait of that character is present widely, and it can be considered as a dominant trait. Okay. And on the other side, the recessive character is a one that basically is like dominated by the dominant character. So that would be less in number. And if that would be the case, that would be fit. The dominant character would be fit than the recessive character. That's why the recessive character is being suppressed by the dominant one. So, am I clear, or like if I have understood your question correctly? Okay, so let's move on to the next question for now. Yes, recessive character is suppressed because it will not be able to like express itself once the dominant character is present along with it. So let's move on to the next question. In paternity case, a single mother claimed that a certain man was the father of her baby. The man denied it, claiming that her current boyfriend was the father. The court ordered a blood test that is much cheaper than the DNA testing to see if he could be ruled out as the father. The mother has a blood type O, the baby has a blood type O, and the man was type AB. Is it possible that he was the father? So, what can be the correct logical answer? Okay, any more answers? Okay. So the correct logical explanation would be if we see this is the male, this is the female. Okay. So in case of the female, the blood group is type O. So the genotype would be small I and small I. Okay. From these two, they have a baby. So in that case, the blood type is also O. That is small I, small I. Okay, because the O blood type is a recessive character. Okay, so for a recessive character to be expressed, the male would also be type O, that is small I, small I, right? But the man in question is type AB. Okay, that means it has gene for A and gene for B both. Okay, so if this is the case, then the child would be either A or B, but not O. Okay, so that's why the person in question is not the father of that particular child. So the correct answer would be option number two. So, do you have any doubts in this particular question? Yes, the father would have the blood type that is O. Okay, so let's move on to the next question. A phenotypically normal fruit fly was crossed to another fly whose phenotype was not recorded. If of the progeny, 3 by 8 were wild type, 3 by 8 were ebony body colored, 1 by 8 had vestigial wings, and 1 by 8 had ebony body color and vestigial wings. So ebony body color and vestigial wings are recessive characters, and their genes are located on two different autosomes. So this is not a linkage question at all. Based on the information, which of the following is likely the genotype of the parents? So what can be the correct answer? Okay, any more answers? Okay, so here we'll consider wild type as E positive for the body color and VG positive for the body color. Oh, sorry, wings. Okay, so 3 by 8 were wild type. That means this was the genotype of the parent for one of the offsprings. Then the other one was ebony colored. Because it's recessive, we'll write small e, and body color was uh, here in this case it was wild type. Okay, so that would be VG plus. Okay, then the third one was vestigial wings, but the body color was wild type. Okay, so that would be E plus, E plus, and then it would have vestigial wings. So VG and VG, because this is a recessive character. And the last one was ebony body color and vestigial wings. So both of them were small e and VG VG. Okay, so if one of the parents is heterozygous and the other one is homozygous, okay, so then, or both of the parents are heterozygous for both of the genes, then only this kind of phenotype would be observed. Okay, because E positive is dominant and VG positive is also dominant. Okay, so if third case would be there, then there would be no, for example, if we see for E positive only, so there would be no wild type, no ebony body colored offspring would be observed. Okay, because in this case it is heterozygous, but in this case it's homozygous. Okay, so because we are observing ebony colored offspring also, so this is actually ruled out. Okay, so, for these offsprings to be observed, both of the parents must be heterozygous for both of the offsprings, for both of the genes. Okay, then only we'll be able to observe these kind of offsprings. So, the correct answer would be either fourth or second. Okay, so is the explanation clear? Fourth option will also be correct. Okay. So let's move on to the next question. In endosymbiont theory, this explains all of the following except. So basically, the gametes would be different. Okay. So for example, if this particular genotype is there, then two types of gametes will be formed, either E positive or E, like, okay. So this is the wild type color and this is the ebony body color. And similar is the case with the wings. So this is vestigial, this is wild type, and this is the vestigial wings. Okay. So in this case also two types of gametes would be formed. This is Vg positive and this is Vg, that is the vestigial wings. Okay. So likewise, if we draw the combinations, then only the recessive phenotype would be formed. Okay. That's why either the parents are heterozygous for both the loci, or one would be heterozygous or the other one would be homozygous recessive for the both the loci. Okay. So these can be the case. So what can be the answer for this particular question? Okay, any more answers? Okay. So endosymbiont theory basically explains how the mitochondria and chloroplast originated in the eukaryotic system. Okay. So in these particular options, option 1 is the presence of mitochondria in ribosome. I think it would be the other way around. Okay. Or else consider like this. Then the presence of mitochondrial DNA, yes, this can be explained by the endosymbiont theory. The presence of membrane composed of the lipid bilayer in chloroplast, this can also be considered as an endosymbiont theory postulate. And the presence of membrane protein on the prokaryotic cell membrane. Okay, so endosymbiont theory does not explain this. Okay, and neither it explains the first option. So the correct answer would be option 1 and 4, both. 1 also. Okay, so the endosymbiont theory doesn't explain these two options. Other options can be explained by the endosymbiont theory. So is the explanation correct?
Sir, if we observe the first option, see that the presence of mitochondria in ribosomes means I did not get that point actually.
Okay.
So ribosomes are present in mitochondria, na? The sentence may not be correct. I think
Yes, yes,
so.
yes.
Yeah,
Because the
should I
option was written
stop
like
it?
that, that's why I included it in the option. But if the sentence would be like, if the presence of ribosomes in the mitochondria, then that option would also be
Yeah.
correct.
Okay. Thank you.
Yeah. In the last option, it states that the presence of membrane protein on the prokaryotic cell membrane. Okay. So the endosymbiont theory basically explains that how a mitochondrial chloroplast basically evolved and how it entered a eukaryotic system. Okay, because they were considered as a prokaryotic system, but as the evolution happened, they are engulfed by the eukaryotes and they started acting as an organelle in an eukaryotic system. Okay, but the endosymbiont theory doesn't explain anything about the prokaryotic system and how it originated. So that's why fourth option is neglected, and it will not be able to explain that thing. So am I clear?
Yes, sir. I have one more doubt there.
Yes.
Actually, in for example, if we take plant cell, it has both mitochondria and chloroplast, right?
Hmm.
So if we consider that cell is evolved from two prokaryotic cells, is
Yeah,
it true?
yeah, we can consider that. Because in case of plant cells, what happens is the number of mitochondria is very less. Okay, because as we see the evolution, first the plants came, then from the plants the animals came. Like the first cell was cyanobacteria. That's what the basic evolution pattern explains. The photosynthetic organism first was cyanobacteria, right? So in cyanobacteria we have the chloroplast, but from there the other organelle also like developed, which doesn't play role with the light. Okay, and it helps in consuming that particular oxygen that is being produced. That is the role of the mitochondria basically. Okay, so from there the animal cell originated. So is the explanation clear?
Okay, sir.
Hmm.
Sir, endosymbiont theory explains how the eukaryotic cell is evaluated from prokaryotic cell. Is
No,
it, sir?
no. The basic evolution of eukaryotes is not explained by the endosymbiont theory. It just suggests that two organelles, chloroplast and mitochondria, are small prokaryotic organisms that are living as a symbiont in an eukaryotic cell. Okay, so they do not explain anything about the eukaryotic evolution.
Okay. Okay, sir. Thank you.
Sure. So let's move on to the next question. Suppose that the surface area of an erythrocyte is 167 micrometer square. Also suppose that the membrane of this erythrocyte contains 4.17 into 10 to the power minus 16 mole of phospholipids and 3.1 into 10 to the power minus 16 mole of cholesterol. The cross-sectional area of a phospholipid molecule is zero point 0.7 nanometer square, and that of the cholesterol is 0.38 nanometer square. Phospholipid and cholesterol isolated from this membrane were spread onto a monolayer. The ratio of the area of this monolayer is to that of the erythrocyte is approximately how much? I know the question is too complex. You write it down, then try to solve it. So monolayer is basically a layer in which only a lipid is arranged. Okay, like we have the cell membrane, that monolayer is arranged in the form of a lipid layer in vitro system. Okay, so that doesn't have any other, like the cell membrane is bilayer, right? And one of the leaflet of that particular layer will be called as a monolayer. Okay, hello. Okay, so let's solve this. Correct answer is option 3, that is 1.5, and this is the solution. Okay, so because there is mole, So 1 mole consists of 6.023 into 10 to the power 23 molecules. Okay, that is the Avogadro number. And because there are 4.15 into 10 to the power 9 minus 16 phospholipid molecules, that yields this much molecules of phospholipid in this particular membrane. Its area is zero point 0.7 nanometer square. So the total surface area covered by the phospholipid would be this number of molecules into zero point 0.7 micrometer square or nanometer square. Okay, so this would yield 175 micrometer square. Fine. Then same with the cholesterol, its surface area is given 0.38 nanometer square. So this would be the actual surface area that was like covered by the cholesterol molecules in that monolayer. So to form this particular monolayer, these two components were used. So the total surface area from these two components would be added up, and that would be yield as 246 micrometer square. Okay, so the ratio of this monolayer to erythrocyte would be 246 by 167, because the surface area of the erythrocyte membrane is already mentioned here. So that would be 1.5. The ratio would be 1.5. So any doubts in this particular question? Okay, so let's move on to the next question. A normal cell has n amount of DNA in a cell. Okay, so this is a interphase nucleus, you consider this. The amount of DNA remaining in the cell after S phase of the cell cycle would be. So what can be the correct answer? Okay, any more answers? So yes, the correct answer is option 2. Yes. So if we have n number of DNA molecules in a cell and the DNA is replicated, so the number of molecules would be increased. Okay, because the DNA is now two molecules. So the in the S phase, the DNA amount becomes from n to 2n. Okay, so the correct answer is option 2. Any doubts in this question? Okay, if not, then we'll move on to the next question. A normal cell has n amount of DNA in a cell. The amount of DNA remaining after complete meiotic cycle would be. So what can be the correct answer? I think it would be enough, whatever has been taught in the lectures and your this year's quiz. Okay, any more answers? Okay, So what happens in meiosis? First is the, so this was the original amount. Then during the cell cycle S phase, it would turn to 2n. Okay. So from this 2n, first is the meiosis 1, that is equivalent to mitosis. Okay. So that would come n into 2 nucleus. Okay. Then from this n, there would be a second meiotic cycle that would divide these two cells also into other two cells. Okay. So other four cells rather. So from one cell, two cell will be formed, and from this cell also two cell will be formed. So this would be n by 2, n by 2. So after completion of the final meiotic division, the DNA that would be present in all the cells would be n by 2. Okay. So because meiosis gives rise to haploid cells, so that would yield in n by 2 DNA amount in the offsprings. Okay. So the correct answer is option number 3. So any doubts in this particular question? Okay, so let's move on to the next question. So, which of the following statements is true with respect to membrane fluidity of a phospholipid bilayer? So, what can be the correct answer? Okay, any more answers? Okay, okay. So, basically, membrane fatty acids membrane fluidity depends upon various conditions. Okay, so let's see the options. So increasing proportions of long chain fatty acid decrease membrane fluidity. Yes, this is correct, because the presence of long chain fatty acids actually make the membrane more rigid, because they will not be able to flow properly. Okay, so this is a long chain fatty acid, and this is a short one. So if we see this kind of structure, that means this will be more rigid in nature. Okay. Then increase in cholesterol content increases membrane fluidity. No, this is also false, because cholesterol content actually, because it is immovable structure. Okay. So cholesterol decreases the fluidity of the membrane. Increasing proportion of cis unsaturated fatty acid increase membrane fluidity. Yes, this is correct. So, in terms of cis fatty acid, what we mean is the bonds will be like this. Okay, because these two chains would be on the same side, so this is called as a cis bonding. So, if we have this kind of structure in a lipid, that means this kink will be formed, and if this kink would be there, then that kink actually increases the membrane fluidity. So this option is also correct. Increasing proportion of trans unsaturated fatty acid increases the membrane fluidity. No, this is false, because in that case what will happen, if this particular This would be like this. So this would interfere with the next phospholipid molecule that is present. So this would actually hamper the interactions in the membrane. So this would decrease the membrane fluidity. So the correct options would be P and R. Okay, so this second option is the P and R. That would be the correct answer. Okay, so any doubts in this particular question? Okay, so this was the last question for today. So if you guys have any doubt, you can ask. Okay, if there are no doubts, then we'll close the session for today. Thank you guys for joining in and all the best for your exams. Actually, I don't know the type of the exam, but it will contain some MCQ questions and some subjective answer type might be there. So you get prepared for that thing. Okay. Mm. Thank you guys. All the best for the exam.